Hello, everybody. In this lecture, we're going to talk about how to recharge ATP. So let's get right into it. The objectives for this are to be able to explain what oxidation reduction reactions are and why they're considered coupled. Also, list the general mechanisms for phosphorylating ADP to ATP. So if we start out and just do a quick overview of redox, these are oxidation reduction reactions. And oxidation is going to be a loss of electrons, while reduction is a gain of electrons. And so if you use this mnemonic oil rig, oxidation is loss, reduction is gain, you get a good basic understanding of how to think of a redox reaction. So to take that a step further, we have reducing agents and oxidizing agents. And so the reducing agent is going to be the electron donor. And then the oxidizing agent is going to be the electron acceptor. Now, what that means is that the reducing agent starts with extra electrons, and then it ends in an oxidized form. Now, the oxidizing agent starts with fewer electrons, and then it is reduced to then gain electrons. All right. And so it's a little bit confusing because you have these names kind of flip-flop, but I always like to keep in mind that oil rig, right? So whatever is at the end, so oxidation is loss. And so who lost electrons? Well, A lost these electrons. Then reduction is gain. Who gained electrons? B gained the electrons. All right. And so these redox reactions are going to be used by cells to get energy, and they do this by oxidizing nutrients. And these nutrients are generally going to be and contain a lot of hydrocarbon bonds. So these are those CH bonds, and these are highly reduced bonds. And so when we oxidize them, they actually release energy. And so your sugars and your fats are going to be the most reduced, and your proteins are also fairly reduced, but not as much as sugars and fats. And what that means is that your cells are going to generally use those carbohydrates and fats first, and then they turn to proteins. And this, like, as a little like pop culture aside, this is kind of the basis behind things like the keto diet, is that you stop eating the carbohydrates and or you stop eating carbohydrates primarily, which causes your cells to focus on the fats and to some extent the proteins as well. And that is where you get the ketosis from. All right, so we have coenzymes involved in redox reactions. They're actually quite important. And so in all these different metabolic pathways that break down all of the different nutrients, you're going to have things like NAD plus and FAD that are going to be these electron acceptors, right? So they're these sponges that suck up those extra electrons, and the transfer of those electrons is what can ultimately generate energy. And so here we have NADH and its reduced form of NAD plus. And why this actually listed as NADH plus, I'm not sure. But NADH and NAD plus are going to be the things that we use to do a lot of oxidation reduction reactions or redox reactions. And so as you transfer electrons, you're going to get this this switching back and forth between these two forms. And we'll talk more about that when we get into glycolysis and the Krebs cycle. But this can also happen with FAD and FADH2. And so when we have these coenzymes, they're going to be tightly tied to different catabolic pathways that are going to cause the phosphorylation of ADP to ATP. So phosphorylation is that gaining of phosphorus. And so if you have an ADP and then you add a phosphorus, that's going to make your ATP. And so again, we're going to talk more about how that happens when we get into the specific metabolic pathways. But one of the most effective ways of doing that is by using these coenzymes like NAD plus and FAD. Now, there's also something known as substrate level phosphorylation. At substrate level phosphorylation, you're going to have an enzyme that actually transfers the phosphate from some sort of molecule directly onto your ADP to make ATP. So in glycolysis, for example, we can transfer the phosphate from phosphoenolpyruvate onto ADP to make ATP. Now, these phosphorylated substrates like this phosphoenolpyruvate are considered to be high energy intermediates. And so that means that they have a lot of potential energy in them and can transfer over to your ATP to make that, molecular currency. Then we have oxidative phosphorylation. This is going to be a bunch of different redox reactions coupled together. And this is going to be part of respiratory systems, both aerobic and anaerobic. And as you see through here, we have our NADH to NAD plus. Then we have succinate to fumarate, so forth and so on. And all along here, what's happening is we're actually transferring electrons through this chain. I do not expect you to necessarily know all the pieces of this chain, but understand that what's happening is we're transferring electrons through here. And as we're doing it, we're pumping out protons. And ultimately, as those protons are pumped through this pump right here, it makes new ATP, which we see right there. Now, as I was saying, this is going to rely on redox reactions. In your oxidative phosphorylation, it's also going to do the same with our photophosphorylation. Now, in this case, the difference is that you're using light to activate those electrons and use those to be pumped through your ATP synthase. And that is going to result in changing ADP to ATP. Now, the big important thing here is that the only organisms that do this are organisms that are photosynthetic. All right, and so that's a brief overview of redox and how to different ways that you can phosphorylate ADP to ATP. We're going to talk about some of that stuff specifically in the upcoming lectures. I hope you enjoyed. What I think you should think about doing before you come to class is write down 1 or 2 questions about these topics that you would like to have clarity on that maybe weren't as clear during the presentation. And when you come to class, we can talk about it a little bit before we get into our questions. All right. Have a good day.